Hello and welcome to this Pharmaron podcast, part of our DMPK Insights podcast series. My name is Simon Taylor. I lead the DMPK function within Pharmaron's Integrated Drug Discovery Department, and I'm delighted today to be joined by Kevin Beaumont.
Hello, Simon.
Kevin has worked in the pharmaceutical industry for 37 years in the fields of drug metabolism and pharmacokinetics. His major area of expertise is in the modulation of physicochemistry to affect drug disposition and in the prediction of human pharmacokinetics from preclinical information. Kevin joined the Department of Drug Metabolism in Pfizer's Sandwich site in the UK in 1983. Under the mentorship of Dr. Dennis Smith, his early career involved developing a fundamental understanding of the basics of DMPK support to discovery and development projects. This included developing expertise in small molecule bioanalysis, completion of radiolabeled ADME studies, and direct DMPK support to discovery and development projects. In 2011, Kevin transferred to Pfizer in Boston, Massachusetts, where he was responsible for DMPK and ADME support to a variety of therapeutic areas across cardiovascular, inflammation, immunology, and rare diseases. In 2020, Kevin returned to the UK and is currently Director of DMPK with AstraZeneca, supporting oncology R&D with DMPK expertise, including across the various modalities. He is author of over 50 peer-reviewed publications across a range of topics, strategies, and opinion pieces relating to DMPK in discovery and development. So, Kevin, welcome. I mean, the theme of today's podcast is the role of the DMPK Project Representative, and different companies have different names for this role, but this is the person who essentially devises and coordinates the strategic input to a project, working not just within their own discipline, but also integrating DMPK science across functions to help design compounds with optimum properties, mitigate risks in further development, and work across functions, pharmacology, toxicology, formulation, and clinical, where DMPK science is often the driving force. And of course, we're probably biased, but maybe we'd argue it's the most exciting role in a project team. And today in this podcast, it'd be great to walk through your career journey, explore the path you've taken, what's changed in DMPK in both drug discovery and development science and strategy during that time, and what you've both contributed and learned throughout your career. So, Kevin, let's go back to the start to 1983 when you joined Pfizer. How did you get to join the organization? What was your first role?
First role was what doesn't exist anymore. It's called a generalist role, DMPK generalist, which was jack of all trades, master of none, basically. So firmly rooted in bioanalysis. So I did a lot of bioanalytical sampling of looking at PK samples and generating PK information, and then as in vitro studies came in, started to generate in vitro data as well, which you do the incubation and then you do the bioanalysis, right?
Yep.
So fundamentally a bioanalytical background, which gives you the in to understanding a lot of the other properties of physicochemistry and extraction efficiency and stuff like that. So, and then how it relates to PK in general. I turned my hand to just about anything, basically.
And I guess that was the role back at the time, wasn't it? Get
the
exposure
role.
to all the different aspect studies.
At all.
What about radiolabel work as well?
We did some I did radiolabel work. Yeah. So looking at metabolism.
Yep.
A lot of urine and feces. I didn't do the mass spec end of it because that was a specialism at the time.
Yeah.
But yeah, 14C studies for sure. But that was back in the development phase, right? We were still, even though we were moving into discovery, you still had to do the development piece.
Yes.
So yeah, it was
so is that
very
still
varied.
Molecule characterization and understanding in development.
Absolutely, yeah.
And was this in a kind of development DMPK environment?
It was a very interesting time for DMPK. I didn't know that at the time, right? So when I started in the industry, DMPK was very much in the development phase, supporting safety, toxicology, and going into the clinic. But it was becoming pretty obvious that if you optimized compounds just on the basis of potency alone, you ended up with compounds in the clinic which were very poor in terms of their pharmacokinetics. And I don't know whether you remember that Kohler and Landis paper from 2004, human PK attrition. Everybody talks about it, right? I use it a hell of a lot in my presentation. That, pre the kind of decade before the 1990, there was something like 40% of development candidate attrition due to poor PK in the clinic. So you'd gone all the way to the clinic, done all the safety, started dosing your healthy volunteers or patients if you were doing oncology, only to find your half-life was an hour, or your bioavailability was poor, or there's huge variability in the PK. So what that drove, in particular, starting at Pfizer, actually, I was very lucky to be in the group that led the charge from development into discovery. An emphasis of working with the chemists to try and optimize chemistry to make sure you pick candidates with the right balance of potency and pharmacokinetics. So that was a great opportunity. And I'll give you one particular example, if you like. I don't know whether you've heard of amlodipine. It was a huge seller for Pfizer back in the day. It's now obviously off patent and generic. But amlodipine was actually discovered in the dog. The dog discovered amlodipine. So essentially, Pfizer were working on calcium channel blockers for antihypertensives, and fast following Bayer, who had discovered and developed nifedipine, which was a short-acting compound, had a half-life of a couple of hours in human, needed to be dosed 3 times a day. Mm-hmm. So what Pfizer were trying to do was make a molecule that would have a once-a-day potential, so a long half-life agent. And what they did was they put a basic center into the scaffold of nifedipine, and the basic center was N-dimethyl. So they had a pKa of probably 9 or 10. And they put that compound into development as a candidate and went and did some tox in the dog. And essentially, on day 1, the dogs are fine. On day 2, they all dropped the blood pressure through the boots. So what happened was, they didn't really know what was going on. Someone must have postulated it was something metabolic, some metabolite or something. And the urine was brought back from the safety group from those dogs. And a guy I think you might know very well, Alan Beresford.
I do know Alan.
From GSK. Yeah. Well, he was at Pfizer at the time. He analyzed those dog urines and found an NH2 metabolite, so 2 demethylations, and that's amlodipine, which was serendipitous because it was the first dihydropyridine calcium antagonist that was actually excreted as the dihydropyridine in urine. And it's only about a 2% metabolite, but he found it, then it was made, and the rest is history, really. So it just goes to show it's all about serendipity. But clearly that showed you that if you have drug metabolism expertise in the discovery phase, that would have been found out very early.
Yes.
You probably could have found amlodipine much earlier and then nominated that instead of the N-dimethyl.
I was at GSK at the time when DMPK also made that transition from development into discovery.
GSK were kind of on the forefront of that and leading in terms of the market, right? So they had ranitidine, that was a kind of first blockbuster, right? So Pfizer and GSK were kind of neck and neck, basically. So.
And how did that transition kind of occur with the discipline into discovery?
So it was tough, right? So you take a group of basically just used to taking one development candidate and working out what the ADME properties of that molecule was in depth, mainly around metabolism and clinical PK, and you transition them into a fast-moving, lots of compounds, high turnover, rapid turnaround area. It needed a different skill set, and that was where I was lucky in that I hadn't learned all of the development skills, so I came straight in and started working on discovery, and it was good. But it needed a real good backing from management, right? That what you're going to do and getting the tools in place to be able to do that. Although I have to say, it was unrecognizable from the tools that we have today. Everything was in vivo. There were no in vitro tools or anything like that. So, but it was again unrecognizable. Chemistry over the corridor, DMPK the other side of the corridor. Your chemist would make the molecule, trip it over to DMPK, and then you could be in dosing. Yeah. Whereas now it's probably 2 or 3 weeks before you get your compound once you've decided to do it.
Maybe it depends where you are.
Yeah.
And so I guess the discipline moved from maybe somewhere in development where it was about understanding the profile of the molecule you have.
Telling the ADME story of the drug
as opposed
a
to,
regulatory submission, essentially.
yeah,
If
and
it got that far.
and it moved to a position of being able to design those compounds. And obviously, you've talked here about your role as a kind of started to be a project rep in the discovery area. I mean, how did you get trained in that role? What was the approach at Pfizer at the time? You talked about starting with small molecule bioanalysis. I had a very similar journey in my career. What kind of concepts and things did you have to start to understand to be able to work in that different environment with med chem and design molecules?
So again, for me, it was serendipitous, right? I learned the skills I had to learn to be able to do that role, and we were kind of learning on the job, if you like. So it literally was understanding chemistry and how, the soft skills are important. Making sure that you could actually talk to your chemists, right, and you could work with them, and you had a good working relationship, and then working together to make sure that you did introduce moieties in the molecule that were like metabolic liabilities. But as you well know, that there's an organization called the DMDG that was instrumental in, and still is instrumental in, learning those skills, right? So I went on the fundamentals course, and that was an eye opener for me. I learned a massive amount, and you bring that back and you apply it to your day-to-day role. The bioanalysis course went on. The PK course was absolutely fundamental, understanding PK. So you learn as you go along, and you take advantage of all of the training opportunities, and you work with people like Alan Beresford and Paul McRae at the time in Mass Spec, who've done this job and done it very successfully. So,
and I guess there's other concepts that are really important. Maybe we could explore some of those kind of important concepts that have kind of emerged or become of increasing importance and it really become part of fundamental DMPK understanding. In the opening remarks, you talked about your interest about the relationship between physicochemical properties and the application to PK. And knowing Pfizer in particular, there was obviously the publication by Chris Lipinski and other colleagues in 97 around that drug-like space. Maybe we could make a comment on how
before we go there, I would say again, serendipity. I was looking for Pfizer. Nobody knew what who Pfizer were, right? It was actually quite a small pharmaceutical company at the time, would you believe? And you say, I'm working at Pfizer in Sandwich. Oh, Phisons? Phisons were the fertilizer company, which then became Astra, right? And then AstraZeneca. And also working at Phisons Pharmaceuticals in Loughborough, Charnwood, was a guy called Dennis Smith, which I'm sure everybody's heard of. Again, the Phisons group were kind of stuck in development. They weren't doing the discovery stuff, and I think Dennis really wanted to do the discovery stuff because he saw the impact it could have. And I was fortunate that he moved from Phisons, having been very successful at Phisons, and came down to Sandwich. Now the guy is phenomenal. It was phenomenal as a leader, a genius in terms of DMPK, and he obviously saw something in me and gave me opportunities to do this sort of, and a lot of this stuff was around that time that Lipinski was coming up with the rule of 5 mnemonic, as they called it. Interesting, right? Because he was a Pfizer colleague, right? US Pfizer colleague in Groton, the big site in the US for research for Pfizer, and he came up with these rules for ADME almost. But what Dennis was able to do was interact with Chris and bring him over to the UK for visits and make presentations. And we worked closely with Chris Lipinski and Franco Lombardo and Bill Domini and those other authors on that paper that never get mentioned. And it really became, and chemistry at Sandwich really took on board the optimization of physics, mainly driven by, again, Dennis Smith collaborating with Simon Campbell, the head of chemistry at Pfizer Sandwich at the time, and they embedded the whole thing into design. And it became very successful. And then you could, and I'm part of that. And I've got these success stories to go out to DMDG meetings and other meetings to start talking about it. Dennis was always very keen on, you do good science, you present it, and you publish it. He was a stickler for that. And that's why I've got 50 odd publications. Same time we had Barry Jones in that group. I don't know whether you remember Don Walker.
Yeah.
Paul Morgan, and it was all mentored by Dennis. And you can see the impact of that across the industry in terms of DMPK now. All four of those guys became I would say, very successful DMPK. I mean, Barry works in your organization now, right? So
he does. Yeah. And I mean, those guidelines were embedded across.
Seminal and spawned a very successful 2 decades of small molecule drug discovery and development. I think they're kind of getting outdated now, and we can talk about that.
And that's, I guess that's where the next question was going. There's a lot of activity now in the so-called beyond Rule of 5 space. Of course, it's only called beyond Rule of 5 because of the guidelines before. How do you see, I guess, them being relevant today?
Yeah. So the reason it was so successful, you remember that, again, back to that Kohler and Landis paper, the 40% attrition due to PK came down to like 10% attrition. And that was purely the application of PhysChem into compound design. And it was an idea of its time because you could do it. You could balance the physicochemistry within Rule of 5 and still get potent agents because the physicochemistry of the targets allowed it. When you're working on aminergic DPCRs where you're building off ligands at a 300 molecular weight, adding lipophilicity and weight still to get antagonists that are potent still maintains you within Rule of 5 space. And you see all of the kinases, you see 350 molecular weight kinases, they're kinase inhibitors. You can have molecules that are extremely potent and still are but within Rule of 5 and are absorbed and have decent clearance, etc., etc. I think that most of those targets have been mined out, and now when you're kind of faced with where do I work with respect to pharmacology and target selection, and you're seeing PROTACs coming through, degraders. Well, degraders by their nature cannot be below 500 molecular weight because they need the point of interaction, they need a linker, and they need the E3 ligase. And so they're all going to be 700, 800 molecular weight, and they're all going to be big and greasy and non-Rule of 5 compliant. And some of so there was a move throughout the decades of the Lipinski-ism, if you like, to call drugs intractable. They're called drug targets intractable. And they're only intractable because you couldn't get Rule of 5 compliant ligands for them. Now we're having to go after some of those intractable targets because they are compelling targets for the diseases we're interested in, oncology, cardiovascular, etc., etc. So you're having to work outside of Rule of 5 space. Now, Lipinski never said you cannot work outside of Rule of 5 space. His analysis was a 90% rule, right? So 10% of drugs are outside of his space. But it's much more difficult to balance the properties that you need for good PK and potency outside a Rule of 5 space. And that's the beyond Rule of 5 situation that we find ourselves in. Me as a DMPK rep for AstraZeneca working predominantly on above 500 molecular weight, lipophilicity through the roof. And you have to balance that and you have to make compromises. And that's, I think, where if you doggedly follow we're only going to nominate compounds that have perfect PK, you're never going to have candidates. You're going to have to take some risk.
Yeah, maybe we'll explore that later.
Yeah.
I mean, maybe just touching on a couple of other key concepts that have probably emerged. Free drug hypothesis.
Yeah, I got a few stories on that one, right? So Dennis Smith. Yeah, I'll always go back to Dennis, right? He's my mentor. He's the reason I'm here, and he gave me my career as it is. So I'm always very grateful to him for that 20 years or so we had together. So I was doing my generalist role, right? We're working on antimuscarinics for irritable bowel syndrome and urinary incontinence. And we had a molecule which we call zamifenicin, which, having talked about optimizing PhysChem, it was a horrible molecule. It was greater than 5 log P. So it was against the Rule of 5. But it was absorbed and it worked. But it was, because it was so lipophilic, it was turned over really rapidly. So in animals, clearance was through the roof. But we took it anyway because in our human prediction from in vitro systems it looked pretty reasonable. But we expected it to be pretty high clearance and lowish bioavailability. But because it was potent, we could probably get away with it. Okay. So I was actually doing the first in human bioanalysis. So I had my HPLC UV assay. So this must have been late mid 1990s. HPLC UV mass spec hadn't really come in by then. HPLC fluorescence, I think it was. Anyway, and the first in human was done at a centre in Plymouth called the Diving Diseases Research Institute. I remember it to this day. All right, and we started our starting dose was 1 milligram, and our predicted efficacious dose was 100, 200 milligrams. We were fully expecting to see nothing in this plasma. Low dose, low bioavailability. I've got a limit of quantification of 2 nanograms per mil, which was a fantastic assay in those days. Yeah. Using 2 mils of plasma as well, and extracting things. Right? No one extracts things anymore, do they? Just lob methanol in or something. So expecting to see this, and wanted to get good rapid turnaround. So the samples were couriered up to me in Sandwich from Plymouth, and I started extracting them in the morning. Took all day to extract them. Put them on the autosampler. Like I said, expecting to see nothing. I thought I'd just stay to see the first few off, right? First sample came off, and there was a peak where zamifenicin was, which corresponded to about 20 nanograms per mil. And I knew this thing there should be nothing, right? So, what the hell have I done? I've mixed up the samples. It's a standard, I think. And I thought I'd better stay and see what happens. And as the samples came out, it was looking like proper PK.
Good profile.
Up and down, it was a decent profile. And it was really zamifenicin in there. So we've got this 1 mg dose that's giving us exposure that would be potentially efficacious if everything was correct. I mean, we figured out I hadn't done anything wrong. Got the boss in the next day, and I said, look, there's zamifenicin in these samples, pretty high concentration. So that caused a stir, got the clinicians in, got the clin farm people in. I phoned up the guy at Diving Disease Research Institute and said, we think you've dosed wrongly. You've overdosed. We think you've dosed 100 mgs and you should have dosed 1 mg. I said, no, I haven't. I'm absolutely certain we definitely have not done that. And just to prove it to you, I'll bring the residues over and you can analyze them. So he's a pilot, an amateur pilot. So he got in his plane, taking the residues with him, and he flew it over from Plymouth Airport all the way over to Manston Airport, which is a few miles north of Sandwich site. And they analysed, in analytical research, they analysed the residues, and there was indeed 1 milligram in those dose residues. So we had to get our thinking caps on. Well, why the hell have we got 20 nanograms per mil when we're expecting less than 1 nanogram per mil? And it was Dennis that figured it out. Right? So basically, he said, well, I think what you've got is a big difference in plasma protein binding. So what you've got is in animals, you've got a certain amount of protein binding. It was high, above 99%, but in human, it's more highly bound. So for the same intrinsic clearance, you're going to get higher total drug in human than in animals. After about a month of me trying to investigate, working out what the plasma protein binding of lamifenicin was, because all we'd done was quantify it to greater than 99%.
Yeah.
So I had to get a really sensitive assay for the free. It was 99.8% bound in rat, 99.98% bound in human. Now that doesn't sound a big difference, does it? That's a 20-fold difference in the fraction unbound. So what that means is, for the same intrinsic clearance, roughly, the molecules had much higher total concentrations in human than in animals. And when you corrected for the total down to bring back the free, which was unheard of in those days, you got the same free concentration, but obviously at different doses. And one of the things with antimuscarinics is there's a translational biomarker, which is pupil dilation.
Okay.
And that happened at exactly the same free concentration in both rat and human. So that meant the free concentrations, not the total, were driving the efficacy or the side effect.
Yeah, that's a really nice example.
And that was the free drug hypothesis, fully embedded in both Dennis and myself's psyche. Yeah. After that.
Yeah. So let's move on. I know you mentioned earlier, you talked about progressing drugs when we discussed the discovery development shift of DMPK, talked about progressing drugs being a real balance of designing properties in. This is often having to be done within an appropriate time frame and probably budget as well. And you mentioned about compromise of properties, and I guess we talk about that balance of risk. When are risks too great in development versus when is it appropriate to push a molecule forward? What's your experience of approaching that balance of risk? When is it appropriate to take those risks?
At the end of the day, it depends on what the risk is, right?
Yes.
So we'll talk probably later about human PK prediction, which is another massive aspect of a DMPK project lead. And so basically, we've developed methodology to be able to help us predict PK in human.
Yeah.
And it's become almost like DMPK can predict PK totally accurately all of the time. A total myth, right? So even the best metrics are we're plus and minus twofold 70% of the time of half-life and whatever. And I've had some spectacular failures in human PK predictions. So trying to drive to a situation where you have a number for PK prediction and then saying that is what the clearance or the half-life of this molecule will be, and then basing your decision on that is naive at best. And sometimes you can't make that prediction because of the situation. So to me, the prediction is there to guide you in making sure you're not doing something absolutely stupid. If I predict a 1 hour half-life and I want it to be once a day, don't take that molecule. That's too high of a risk. If I predict an 8 hour half-life, it could be 4, it could be 16. And if that's the best compound you have, that's worth taking the risk and go and find out what the human PK is. But don't say it's only 8 and I want to get a 24. So I've got to do some more chemistry to go. You see what I mean? So I would take risks with PK. Now, if you're not careful, you can get into a mindset of, I don't want to be wrong, right?
Mm.
And the night before you're getting the results of a first in human PK is very stressful as a DMPK lead, because it's about your prediction and how close have you been. And my range of prediction is from 0% error, which is spot on, to something like 3000%, right? And then you look stupid. Now, if you're not prepared to be stupid and look stupid, and having taken that risk, you end up being really conservative and you never take anything because there'll always be something. So to me, it's about what are you risking? If I'm predicting something that has a chance of being reasonable PK, then I'm happy to go and find out. What you're doing is testing a hypothesis. That's what we're supposed to do.
Yeah.
It's not a fact.
Human PK predictions are really interesting topic for me as well. And one of the things the industry has looked at to try and perhaps de-risk that is microdosing.
Yeah.
I've
done some microdose, a couple of microdoses.
Yeah.
I have to say they're very unpopular within a metrics-led large organization.
Okay.
Because it's perceived that you're spending money and time to go and find out something that DMPK can already predict. Well, the answer is we can't. Right? So a classic example for me was, back in the Pfizer days, we had a molecule that predicted fantastically well from in vitro assays, but if you predicted from rat, you got a bioavailability of like 70%, and you predicted from dog, it's like 0. So you have a range of prediction of 0 to 75%. So what do you do in that situation? So you got 3 options, right? You can take it and find out. And that's what microdose and pharmacologically active doses can do for you, doing it at less cost. Or you can do some more chemistry to find a molecule that brings those predictions together. Or you can just dump it and go and do something else. In that situation, the company weren't confident that they wanted to take it forward in a full first in human. So they decided that they wanted DMPK to do some more work to rationalize the prediction, and they wanted to do more chemistry to find a molecule that didn't have this disconnect, if you like. So we spent 18 months of chemistry, 2 chemistry teams, costed at something like $3.5 million. Expensive stuff, right?
Yep.
We radio labeled the molecule, stuck it in dog, trying to figure out what else was being metabolized, etc., and was that like to be in human. We did more species, but all that did was widen the PK prediction range rather than narrow it. And ultimately, they never find a molecule that's any better. So they decided to do a pharmacologically active dose. It took 6 months to do. And we had human PK. Had a 30-hour half-life and an estimated bioavailability of 80%. It cost about $500,000, which was 1/5 of what we spent on chemistry trying to find it. So you see, it's not cost negative to do a microdose. You have certainty in an area of uncertainty. And maybe that's something we should be doing in beyond rule of 5 space, because we've based our prediction capacity off rule of 5 compliant compounds. And we got this massive back catalogue of predictions on rule of 5 compounds. And we're now working on 700 molecular weight. We haven't got any back catalogue, got no clue. So how are you going to get that history if you don't take these molecules forward based on your predictions? Just my hobby horse. I would do more microdoses, and
you would do more
a lot more.
Yeah, it's potentially something that could
But it's so
really help to
powerful.
de-risk
At compound ID, candidate ID, you'd know the PK. Now people talk about, oh, it could be nonlinear. Well, if you do it IV, it won't be nonlinear, right? And you'll have the clearance and you'll have the bioavailability. You'll have the volume of distribution, all that stuff. You'll know it. So why not? But the bean counters don't like it. Costs money and time.
So we're talking about attrition here as well, kind of minimizing attrition. And one of another famous publication that came out of Pfizer was, of course, around the 3 pillars initiative, ensuring there's exposure at the target, there's engagement, pharmacology.
Yeah, I have nothing to do with
in
that
the clinic, and then demonstration that you have activity commensurate with both exposure and target engagement. And of course, there's been follow-ups from other pharmas, the AstraZeneca, 5Rs, etc. And DMPK departments have responded to these attrition analyses by improving understanding projections, putting in assays to mitigate against various aspects of attrition. I guess you worked at Pfizer during that time. You've also worked at AstraZeneca now. What's your experiences on these analyses actually addressing attrition, and how do you think DMPK departments have responded either effectively or ineffectively to
great
aid
question.
in attrition?
So I can give you the history, right? I wasn't involved in the three pillars analysis, Pete van der Graaf and Paul Morgan.
Yeah.
But I can tell you it was fundamental based science looking at across failures and successes. But when you think about it, what we'd done by approving PK attrition just enabled us to fail in phase 2 later. So it was an absolute disaster, right? And now instead of losing 40% of your molecules in the phase 1 early, you lose them in phase 2 later when you've spent hundreds of millions of dollars, right? And because we could optimize PK, the mistake I think that Pfizer made was choosing pharmacological targets based on whether you could get rule of 5 compliant ligands or not. And what that meant is you chose molecules with not very good biology rationale. And Paul Morgan, Pete van der Graaf, and a few clinicians looked at this, and this is not sustainable. You cannot do this, right? You cannot be failing in phase 2, right? So they looked at it and said, what makes the key in in phase 2 is understanding the biology and how you're modulating the biology.
Yes.
Right. And it fundamentally came back down to biology modulation and measuring the biomarkers to show that you'd actually engage the target. Because what we were doing in discovery, as cowboys in discovery, we'd go against the target and not measure any biomarkers and any of the downstream consequences. We just look at efficacy in a model, animal model. And we take it into the clinic, fail in phase 2 and say, well, it failed because it wasn't potent enough, which enables you to go back in and find a more potent one, nominate that. It's like a candidate factory, right? And it met all your metrics, but you end up with no drugs because they all failed in phase 2 because it was the wrong target. So what the 3 pillars did was made sure that you were engaging the target at least and didn't enable that. Well, it wasn't potent enough. If you engaged the target, didn't do anything, failed in phase 2, that was a bad target. And then that started you thinking about, well, what are good targets? And there are 2 things that make a successful drug discovery development program. 2 decisions. One, have you got the right target, pharmacologically wise? If you modulate it, is it safe and does it do anything in the disease you're interested in? And then it's all about getting the right molecule. Very simple, but very difficult to do.
Do you make it sound?
That was the 3 pillars. I have to say, I moved to AstraZeneca, and the emphasis on biomarker modulation is intense, and it's a really good thing. Probably one of the reasons why AZ has been so successful in the last decade or so. Nothing to do with me, but yeah, I think that because it means that the biology is understood to me.
Yeah.
All right.
Yeah.
You are going after the right target, and if you modulate the biomarker, it should do something. If it doesn't, there's no point messing with that target anymore.
I mean, it's interesting to see how industry has, really focused down and embedded these concepts and how things have changed over time. And
there was a lot of money sloshing around in the 90s and 2000s based on some of the successes, and we spent it unwisely, if you like, on targets that could never do anything in the disease. And I've got a 40-year career. I've got 1 drug. It's an industry of failure. It is not easy. And if you prosecute targets that never have a chance of being a drug, you're spending money wastefully.
I think these concepts as well, in my experience, have clearly were demonstrated clinically, but they've also become embedded preclinically as well. And I think it's really aided translational science as well.
And there are whole new disciplines of PK/PD modeling that are fantastic, right? You can, those guys understand the language of mathematics and how it applies to biological systems, and they can really enable, if you do it properly, if you do the right experiments for them, and that's a different discussion about the collaboration with biology, DMPK, and chemistry. And they can tell you what's going on, and they can model what's likely to be going on in the clinic, making some assumptions, but very powerful.
Yeah, and that marriage of kind of the experimental science and the modeling, which has really emerged over the last decade or so.
Yeah, and that feeds into PK and dose projection, right?
Yes.
So I think it's definitely the way to go, but you have to continually keep learning about the physicochemical space and the pharmacology that you're working in.
So we've talked about your career and the science. What about the future? Where do you see
my future? I'm 41 years in. I'm not sure there is much of a future.
the DMPK Project Rep role. Future. Where are we now with this? Where do you think this goes in the future? Different modalities, different levels of input.
Yeah. So again, when I started, everything was small molecule.
Yeah.
The whole portfolio was small molecule. I think it's getting towards in companies now, on big companies, 50-50 small molecules and biologics.
And you see that coming through in the approvals now, don't you?
Yeah, absolutely. Yeah. So as a DMPK rep, that's a challenge, right? Having spent 30 odd years working just on small molecules, then I came back to AstraZeneca and I'm working on ADCs, which are small molecules attached to antibodies. So now I've got to work out, what's the clearance process for antibodies? Why they got such a long half-life? FcRn, recycling, all of that kind of stuff. So that, you have to go back and you have to learn that stuff. Oligonucleotides, people are going to have to learn that, and all the other fantastic new technologies that cell therapy, gene therapy. There are people, AstraZeneca, working on that in DMPK now.
And the considerations are quite different.
And they're having to generate the knowledge as they go along. So it's highly talented people. They'll get there, but it's a bit like Lipinski and learning about Rule of Five, right? In a bigger context.
Where is the regulatory framework with respect to DMPK in these modalities? We still
Again,
influencing that area?
So I think because basically, when you think about it, in terms of regulation of small molecules, it was us that discovered the problems for the regulators to then regulate. And we work with the regulators to make sure that the regulations are appropriate. That's what's being done now with respect to large molecules. You think the Mabel calculation.
Yeah.
That's an interesting one, right? Based on some of the incidents in phase
one trials, yeah.
But it's becoming a bit too conservative, I think, about the starting concentration. And for some of the T cell therapies, they're starting way far too low. It's taking years to escalate, and that will mean that those therapies won't come through, right? Because no company is going to want to take 3 years in phase 1, right? So somehow you go to the worst case, and then you come back from it and work out where can you safely start with those things. Not my real area of expertise, I have to say, but the new generation, we're going to have to be learn all of the background, the DMPK background to all of those therapies, right? So
yeah,
good luck.
Well, it's an exciting future.
Yeah, absolutely.
Yeah.
There will always be a role for a DMPK project representative and all of the people behind it, right? I don't think anytime soon small molecules are going out of business. I think it'll be a balanced portfolio, and you'll need a range of skills, both as the project representative and those folks in the lab that are doing generating the data. You just have to be adaptable to the skills that you have, I think.
So we talked quite a lot about the science and the strategies of DMPK. Maybe we can perhaps finish with talking about the people. You mentioned some of your mentors earlier on, and I know you're passionate about training junior scientists in DMPK as well, not only in your role, but also through the DMDG. Any particular people you'd like to mention or examples?
Let's step back a bit. I received fantastic mentorship from not just Dennis, but other colleagues within Pfizer, and actually through the DMDG, colleagues that weren't at my company. So having seen that and seeing what effect it had on me, I wanted to make sure that you pass it on, right? Because you're always trying to, mentor the next generation of scientists. So just looking at my career, it was very interesting, right? I said, I was started as this generalist, this sole contributor, started to become a team leader, went to a section lead, up to head of department and all this kind of nonsense. And you lose that, right? You become a manager, not a mentor. And that's what happened with myself, Barry, Don Walker, Paul Morgan. We all became these big group heads that, doing day-to-day management, managing budget, all the boring stuff, right? That you probably know about.
Oh yeah.
And I was thinking, this can't be right. We'd been, the reason we were so successful was because Dennis mentored us in the science, and now we're at this elevated position, and we're bringing all these new people in, and nobody's mentoring them. So I basically went to my boss at the time, which by then it wasn't Dennis, it was Rob Polzer, I think, and said, "Look, I think this is wrong. You're not fostering the next generation of talent. We're just looking at I had 30 direct reports at one point, which is crazy." So I said to my boss at the time, it may even have been Paul Morgan, I don't know. I said, "Look, we're not mentoring the next generation. I want to do that. So their credit, whether it was Paul or Rob, they said, right, we're going to create a position as a DMPK project lead. So you're going to go back and do project lead, project stuff at a high enough, senior enough level, so that you don't have to make a pay cut. And you'll be responsible for bringing on that next generation. And I hope I've done that ever since. And I've mentored lots of folks that have gone on to become very much more successful than I. So, I don't know, people like Charlotte Allerton is now head of early development at Pfizer. Rob Webster, senior vice president in biologics again at Pfizer. And Alison Betts is, I think, global head of ADME at Takeda now. So you give these folks a bit of a start, and then they bring through their own talent, and then they've gone on to do fantastic things. And I'm proud of that. I think that's if I've got one drug, but lots of people have done very well, right? So despite actually reporting to me and being mentored by me, they've gone on to have very successful careers, if you like. So,
and it's interesting in this whole training piece. You talked about being a generalist, being able to see that it's perhaps the landscape these days is tougher for people to do that, and so actually,
well, they can't see everything, can they? We're now compartmentalized into you do metabolism, you do PK, you do, and it's very difficult to see the overview that you need in order to be able to represent your department, your discipline, to a project team.
Kevin, it's been fascinating and insightful conversation today. Thank you very much for your time. We've had quite a wide range of discussion.
It's flown by, right?
It has. Thank you all for listening to this episode of Pharmaron's DMPK Insights series. We'd like to remind you that our DMPK webinar series is also available on demand, covering a variety of key questions, issues, and interviews with key opinion leaders relating to DMPK science in drug discovery and development. Stay tuned for more podcasts in our Pharmaron DMPK Insights series. Thank you and bye for now.